not on projects relevant to the topics that I will be talking about today. Okay. Now, the reason I personally got interested in bacteria many years ago is that they're easily the most dominant form of reliving life on Earth. There is an estimated 1 billion species, and there are estimates saying that there might be 10 times as many bacterial cells in a human body as human cells. And that number has undergone various revisions now. Somebody says it's 1.5 times, somebody says it's 10 times, it's okay. But the fact, though, is that we have more bacterial cells than human cells. And various estimates suggest that the total number of bacteria on Earth is this very large number, that is 1 followed by 30 zeros. And compared to so many mammals and so many insects on Earth. And there is hardly any habitat on this planet in which you will not find a bacterium. So the fact that this kingdom of life has it dominates so many different environments, starting from the benign conditions that are there in our homes to even conditions like hot springs and Antarctica, and also whatever we throw at it in the lab suggests that this kingdom of life has a variety of genetic mechanisms to adapt to whatever situations that they face. Right. Now, over the years, there have been many ways in which we have studied bacteria. If we go back to the 19th century or so, we've had Louis Pasteur and Robert Koch. Your microbiology books would tell about the kinds of work that they did, including, very importantly, to develop approaches to show that any disease that a person has is caused by an infectious agent, for example, bacteria. And going back further in time, we also know that sometime in the 17th to early 18th century, Leeuwenhoek, this sort of a person who was stitching buttons in a small city in Netherlands, was the person who first probably see bacteria under the microscope. It started from there, and 50, 70 years ago, there's the famous phage group in the US with Max Delbruck, Salvador Luria, Joshua Lederberg, and these people started studying bacteria and viruses of bacteria to try and understand bacterial adaptation. They were able to do experiments in which they could subject bacteria to different stresses and see how the bacterium adapts to these stresses. And here we are talking about times that precede any molecular techniques. And fast forward quite a bit, and in the 1970s, the first genome of any organism was sequenced, and this genome was that of a bacteriophage. I forget the name of this bacteriophage, and this whole genome wasn't sequenced in one go, right? And it was this bacteriophage had 3 genes, and the third and the final gene of this bacteriophage was sequenced in the mid 70s, and for the first time we had the complete genome of any organism. And then later on, the complete genome sequence of the lambda bacteriophage was sequenced, and one thing led to another. Human genome project was announced, and there is a lot of interest in trying to sequence genomes of all kinds of organisms. And in the mid 90s, the genome of a bacterium was sequenced for the first time, and this is the genome of a pathogenic bacterium called Haemophilus influenzae. The genome of the more famous E. coli was sequenced a few years later. Now, why do we want to sequence bacterial genomes? And what kinds of information would we get out of a bacterial genome, or for that matter, any genome. The genome, as you all would know, is a blueprint for everything that this organism is capable of. All right, and it also sets limits on what it is capable of, and also provides the ability for the organism to perform various actions. And to a large extent, not completely, of course, by sequencing the genome, we get a fairly reasonable idea of the kinds of capabilities that the organism harboring the genome will have. It typically it goes a little beyond genomes to know exactly what a bacterium or any other organism is capable of doing. But the genome itself, as a static object, is something that gives us a fairly good idea of how these work out. Now, here are some statistics that I picked up from just this morning from the Joint Genome Institute in the US, which contains a database of various genomes, including bacterial genomes. And most of the genomes that we have here would be bacterial genomes. Now, the first genome, as we mentioned, came sometime in the mid 1990s, and over time, the number of sequences increased fairly slowly here. Okay, and then increased fairly slowly here until about the late 2000s, and then overall there's a sudden increase in the number of genomes that we have sequenced. Now, over this period, this first period where the increase was fairly slow, all the sequencing was carried out using the Sanger sequencing, which is quite expensive and also relatively more laborious to perform. And at this point, what we call as deep sequencing or next generation sequencing technologies became available. Now, what these sequencing technologies allowed us to do is to sequence bacterial genomes very, very quickly. Today, for example, I can sequence 100 or more bacterial genomes in a matter of a couple of days, and at a fraction of the cost that it took to sequence genomes using the older Sanger sequencing techniques. Now, anybody can sequence bacterial genomes fairly quickly and for a few thousand rupees, right? And thanks to that, the number of genomes has gone up. Now, what we have here are genomes which are more or less complete. That is, we have a nearly complete complement of all the genes and all the bases that may be covered, that may be present in this genome. But what has also happened over this period is a much more steeper increase, right, in the number of genomes which are what we would call as draft, right? These genomes are not fully complete, but they're complete enough for us to give a fairly good idea of what this organism is capable of. And why we have these draft genomes instead of fully complete genomes has got to do with the kinds of technologies that we are using here. That many of these deep sequencing technologies produce what are called short sequence reads. Each piece of DNA that I actually sequence is very short, which makes it a little bit more complicated to assemble all these short pieces of DNA into a fully complete genome sequence. So it's most studies today do not attempt to complete the genome, but instead try to have a fairly good idea of what is going on in the organism, and also focus in efforts towards what are called resequencing experiments. What are resequencing experiments? I already have a reference genome that is complete of a representative genome of that species. Now I have various various clinical isolates, all of which belong to the same species. All I'm interested in knowing is what are the variations that this clinical isolate might have compared to the reference genome. A lot of studies focus on such questions, and many of the sequencing projects that perform such experiments belong to this category of draft or more or less incomplete genomes. So this is where I would want to stop talking about the kinds of techniques that we use in genome sequencing projects. So I'll be happy to take some questions at this stage before I move on.
Sir, there are no questions as of now from the participants. I have requested them to drop in the chat box and Q&A.
Okay.
You can proceed, sir. At the end of the session, we can take it.
Okay, so I will go through to the next phase, and then I will stop, because if there are any doubts, I would like to clarify then and there, because as things build up, then things can get a little more complicated. Okay.
Okay, sir. Thank you, sir. I request once again participants to raise questions and drop it in Q&A section if you have now. Thank you. Sir, please proceed, sir.
Okay. Now, said that we've sequenced a large number of genomes, and either these genomes are fully complete or they may be in draft stages. And we've learned many things from these genomes. Let's focus on one thing. We now know, which we would have anyway guessed from the large variety of habitats from which we find bacteria, that these bacteria are very, very diverse. And if they're very diverse in terms of their capabilities, which also means that they're going to be very diverse in terms of the genome content that they have. Now, there are many, many things that one can look at when we're talking about genomes. The simplest thing that one would want to see is how big is a bacterial genome, or how long is a bacterial genome, is measured by the number of bases that it contains. For example, we know that the human genome is about 3 billion bases long. Now, these bacterial genomes have a broad range of sizes. The largest bacterial genome that we have is, I think, about 12 or 13 megabases long. So, this is the largest bacterial genome that we know of. The smallest bacterial genome that we know of is only about 100 kilobases, or 0.1 megabases. So the genome sizes span at least 2 orders of magnitude. And if one were to look at this as a distribution of genome sizes, what you would see here, if I have genome size and number of genomes of a specific size, we see something like that, about 2 MB and about 4 MB. And from these, we also know that if a bacterial genome is smaller than, say, about 1.2 MB or so, it is unlikely to have a free living lifestyle. That means it becomes a symbiont or an endosymbiont of some other host, and is very, very heavily dependent on that host for its survival. This bacterium cannot survive outside that host, in which case you see a bacterium which is probably exchanging nutrients with this host. A classical example that most of you should be familiar with is the mitochondria. Now, mitochondria, we know, or we very, very strongly believe, is a remnant of an ancient bacterium which was probably phagos cell, and it became an endosymbiont of these cells, and now has a very, very degraded genome. And we all know that a mitochondrial genome codes for very, very limited capabilities, and nobody will even think It's just not possible, because for its existence, it requires the host cell. And of course, now the host cell is also dependent on the mitochondrion for a lot of different things. And then you have these fairly large genomes, and these are bacteria which will live in very, very complex habitats, have very, very complex lifestyles. For example, bacteria that live in the soil and produce a wide range of interesting and strange chemicals like antibiotic producers and so on. They have very, very large genomes, and they are also likely to be fairly um, um, flexible in terms of the kinds of environments that they can live in. So, just from something as simple as how long is my genome, I can more or less make some prediction about the kinds of lifestyle that this bacterium might have, right? And now, going from genome size, we want to look at something a little at a little more deeper level of detail. Now, once I have genomes, first thing I know obviously is how long it is. Then, within the genome, I can make predictions of what the genes are, right, and what functions these genes may encode using some annotation procedures. For a certain subset of these genes, I will be able to assign a molecular function, and for many others, I will not know what the gene might be, because I have no other example of a similar gene that has been characterized. But um, to some extent, I can make predictions of the kinds of gene functions that are present in this genome. Now, let's look at this graph. Now, what this tells is, it takes a whole range of functions that are known across bacterial genomes. Okay? And now I take a function, in how many bacterial genomes is this function present. Okay, this is a gene function, in how many bacterial genomes is this present. Or here they've sort of plotted it in the reverse, that is the number of genomes in which this function is absent. And as you can see, this graph has a very, very sharp peak here towards the right. And what this tells is that many gene functions, right, many functions are absent in many bacteria. And then if you look a little closely, I can try and zoom in here. If you look a little closely at this left end, this part, what you see is a small peak. If you look at the red line that these authors have placed, there's a small peak here, which tells you that there are a certain set of functions which are present in a large number of bacteria, and there is a whole space in between where these functions are present in a few or in intermediate numbers of bacteria. So there are certain core functions that define a bacterial genome that are present across a vast range of bacterial genomes, and you would expect these to be typically involved in processes such as translation, transcription, and so on. And then you have this whole range of functions that are there only in few bacteria, not there in anywhere else. So, which means that any given bacterial genome is a repository of a whole variety of novel gene functions. So, the more bacterial genomes we sequence, especially from novel habitats that we haven't explored so much, we are likely to discover new gene functions that may be of interest to various stakeholders. Okay, now at this stage, I want to talk a little bit more about the mechanisms of bacterial evolution. Again, at this stage, I'll be happy to take questions.
Sir, can you hear me?
Yes, I can hear you. Yes.
There is one question from participant.
Okay.
Does a bacterial genome varies from more than 13 MB also?
It's possible. Among the known bacterial genomes, the largest we know is about 13 MB. Right?
Okay.
We don't. Of course, we don't necessarily know that. We haven't. Obviously, we haven't explored every bacterial species that is around. So there is a chance that there may be something that is bigger. But what we know is this is the largest in that range.
Okay, sir. Thank you, sir. Please proceed, sir.
Okay, so we will go back to looking at genome sizes, and these genome sizes, though a very simple number, can tell us quite a few things about how bacterial genomes can evolve. Now, in this very nice graph, what we have are bacterial genomes sizes represented as this number line here, and this is in a log scale. Here is a bacterial genome that's at 100 kilo or zero point 0.1 MB, and here is a bacterial genome that's about 1 MB, and here is a very large bacterial genomes. Now, bacterial genome distribution, as I had mentioned, has two peaks, one at around 2 MB, one here, and one at around 4 MB. Now, let's assume that there is a certain mean bacterial genome size, right, which is, say, in this area somewhere. And then let's start from here and think about how a bacterial genome can grow and how can it become smaller. Now, when a bacterial genome grows in size, it is innovating. That is, it is finding new functions, which very likely gives a selective advantage to this gene. And there are many different processes which can result in these kinds of genome size increases. One is gene duplication, right? Where I have a single copy of a gene, it duplicates, and when it duplicates, I have two copies of the same gene. Now, when I have two copies of this the same gene, one of the two copies is now under weaker selection than the other copy. That is, even though this function may be essential for the survival of this organism, just because I have a second copy, I'm free to experiment with this second copy. If you let's imagine you have a certain gadget, right? And you're very keen in trying to explore this gadget, open it up, and try to do things to it so that you can do something interesting with it. But you don't want to do it because you don't want to lose it. It may be expensive, and it may be something that you really need, and you don't want to change it as well. And then suddenly you get a second um, um, uh, um, uh, copy of this same gadget as a gift or something, and now you're so very curious to know how you can play around with this gadget. Now you would be happy to open up the second copy of this gadget and do things with it, and maybe create a new function out of that same gadget. The same concept applies here. A second copy of a gene can be created, and then mutation forces can work on this second copy, and selection will not penalize any mutations that can happen in this second copy. And eventually, over time, this process of experimentation can produce a new gene with a new function. And now the genome has become a little bigger by making a second copy of a certain gene, and it also has a new function, and there is an innovation out there. A second mechanism by which this happens is through horizontal gene transfer. Now, what do I mean by horizontal gene transfer? Typically, when you have genes being transmitted from one generation to the next, genes are transmitted from parent to a progeny. This is the norm, and this is what we call as vertical descent of genes, vertical transmission of genetic material. Now, bacteria especially, but not limited to bacteria, of course, even eukaryotes can do this, have the ability to acquire new DNA from entities which are not their parents. Now, what could these entities be? One, these could be DNA just floating around in the environment. Certain plasmids can be just taken up from the environment by certain bacteria which have the ability to do so. And secondly, we know of viruses, for viruses of bacteria or bacteriophages, which in many cases can add a copy of the bacterial of the viral DNA into the host cell, and while moving from one bacterial cell to another, a virus may also pack elements, aspects of the bacterial DNA itself, and then transmit that into to other bacteria. So, through viruses, bacteria can acquire DNA, which obviously not directly from a parent, but could be from various other bacteria. And there is also, the textbooks would tell you that there is also the process of conjugation that involves the F pilus, through which a bacterium can inject a piece of DNA into a neighboring bacteria. And again, this neighboring bacteria need not be a progeny or anything of that kind. So these are primarily two key mechanisms by which genomes grow in size. Now you will realize that when I'm talking about horizontal gene transfer, I also have a problem with how I think about a phylogenetic tree. Right? When I look at a phylogenetic tree, I draw something like this. I start from here, and then I talk about divergence, something of this kind, right? Um, and when I draw something of this kind, then I know that here is an ancestor of this, and this person would be the ancestor of these genomes. Now, this phylogenetic tree obviously assumes a classical vertical descent. Now, the moment I have horizontal gene transfer, I might have genes going from, say, this uh, leaf all the way to this. And if I, on this classical phylogenetic tree in blue, I do not capture this relationship at all. This is a very, very divergent bacterium. This is from this, and there is no way for me to represent this kind of a genetic transfer. Now, what therefore horizontal gene transfer tells us is that a bacterial evolution has tree-like aspects, obviously, because there is classical cell division, and there is species structure and so on. That is not going anywhere. But on top of that, what I also have is, as people call, forests or networks, where I have cross interactions going, and these can be directional depending on the source and the recipient of horizontally acquired gene. And you will note that many pathogenic organisms that we know of today, many pathogenic bacteria, the ability of these organisms to cause disease is often a result of genes which are horizontally acquired. Um, the most One of the most famous examples would be cholera toxin, right? The cholera toxin was, over a very, very long time ago, was discovered by an Indian, Shambhunath Day, and sometime in the 90s, it was discovered that the cholera toxin is not something that is indigenous to the cholera bacterium itself, but is in fact introduced into the bacterial or the cholera, Vibrio cholerae genome by a bacteriophage. So this is a horizontally acquired piece of DNA which gives this bacterium the ability to cause a disease. And we will see that in so many different examples. Pick any E. coli that causes diarrhea, and you will find that genes which enable this bacterium to cause disease are horizontally acquired. Salmonella. Pick any, and you will find that this is the case. And yet another example of horizontal gene transfer that is of clinical relevance is the phenomenon of antibiotic resistance. And where we now know that many bacteria that cause disease today, and also those that don't, seem to carry genetic elements that allow this bacterium to resist the action or the toxic effect of a whole range of antibiotics, which is a problem because if everything becomes resistant to antibiotics, how do we treat bacterial infections? In many different bacteria and for many different antibiotics, we now know that the ability to of resistance has arisen through horizontal gene transfer mechanisms. So I will get back to this the second half of this, but before that, just take a look at this little graph. Now here we're going to talk a little bit more about horizontal transfer in action, but I'll have to explain this graph. So this graph is from a 2008 or so paper by, I'm sorry I didn't put that citation here already, in Genome Biology. I think it was in 2007 or 2008. Okay. Now, what these authors did is to take all available E. coli genomes at that time. Now we have several hundred fully sequenced E. coli genomes. At that time, there are about 40. Now they took these E. coli genomes and then asked the following question. Now the smallest E. coli genome is about 4 MB, little, right? And the largest E. coli genome is in the 5.5 MB range. So even at the level of genome sizes and within a single species, right? This is just all our E. coli genomes. I have a range of genome sizes going from 4 megabases to 5.5 megabases. Now 4 megabases would typically correspond to about 4,000 genes, and 5.5 megabases would correspond to about 5,500 genes. Now let's there is a question that one can ask. So I have this diversity in genome sizes. Would this be a consequence of this small bacterial E. coli genome, this 4,000 gene bacterial genome, being a base onto which a few hundred genes can be added on and to become various other E. coli genomes with larger genomes. Now, what do we do? So to do this, I take all these genes that are there in E. coli, and then let me just explain this a little better. Okay. Now, what I can do here is make a small matrix. Okay. Here, along the horizontal line, what I will write are the genomes, right? E. coli 1, okay, E. coli 1, E. coli 2, E. coli 3, and so on. And here I make a list of all the genes, right, that I have across all these E. coli. So I have gene 1, gene 2, gene 3, gene 4, and so on. And then I ask, is gene 1 present in E. coli genome 1? If so, I put a 1. Is it present in E. coli genome 2? No. I put a 0, 1, 1, 0, and so on. I fill this matrix completely. And then I ask two questions. One, how many of these genes are present in all, or 99% or something of E. coli genomes. And that is basically, if from your set theory and so on, you'll realize that this is basically the intersection of all the sets. If I take each of these genome as a set, and in which the elements are the various genes, then what I define as the core genome is the intersection of all these sets. Now, obviously, as I add more genomes, this intersection is likely to become smaller. Now, if the smallest E. coli genome, that is the 4,000 gene E. coli, is the base on which everything else got added, I will see that this intersection is basically this 4,000 genes on which other things got added. And then my Venn diagram there would look something like this. Here is the 4,000 genome E. coli on which I will be adding various other things, right? This is what I would expect if under that hypothesis. Now let's see what the actual data looked like. Yeah, okay. Now what you see here is as I add more and more genomes into this comparison, as this matrix gets wider along the x-axis, the number of genomes that are common to all the genomes that I have considered decreases. And you find that once it hits whatever 30 odd genomes, the common set is only about 2000 genes. Now, here is this interesting species where I have a common or the defining set of genes being less than half the uh, the smallest genome, and which suggests that pretty much all E. coli genomes have undergone dramatic processes of genome expansions. And later studies suggested that this is not all expansions, right? And a lot of these expansions that we see here comes through horizontal gene transfer. But what we also see are gene deletions and so on. And there is a combination of gene deletions and gene expansions in many cases through horizontal gene transfer, which has resulted in this interesting pattern within one species. Now, here I mentioned that there is an expansion process that is coming through horizontal gene transfer, and at the same time, there are also gene deletion process. Now, if I go back here, going back to this nice median bacterial genome, I've looked at how genomes increase in size, and the genomes are also decreasing in size, and this is a process of genome degradation or genome reduction. And again, a classical example of this is the mitochondrion, which is a bacterium and which has undergone severe genome degradation. And here, this is something which operates under what is called a population bottleneck. Okay. Now, I will just take a few minutes to explain this and then give some examples.
So before that, there is one question from
Yes,
participant
please. Yes.
relevant to this one only.
Yes,
How
please.
genome size is measured?
Sorry, genome sizes?
Measured. How it can be measured? The size
Measured.
of the genome. Yes, sir.
When you basically when you sequence the genome, right? You're basically sequencing the more or less the entire genome, and the number of letters that is there in the genome tells you the size of the genome. As simple as that.
Okay, sir. Thank you, sir. Please go ahead, sir.
Okay. So here, let me say what a population bottleneck is. Now, for that, we need to be clear about Darwinian selection. Okay. Okay. So let's say that there is a large population of, say, bacteria, and all of these grow at the same rate. Now there is a mutation that arises in one of these bacteria. So, and that mutation makes this bacterium, now that I is in red, grow slower than all of these other bacteria. And in exponential phase, growth is obviously exponential, and very soon this would become such a minuscule population percentage of this population that it is going to lose out. And this mutation, let's say in this case, was a deletion of a bunch of genes, is not very useful to the bacterium and is in fact detrimental. Now consider a different situation here, um, and let's say in this thing, which became red, by accident happened to escape and entered a different pool, right? This is like a small box, right, where there is no other bacterium. So this is the single bacterium which is there. It is the king of this habitat. There's absolutely nothing that this bacterium has to compete with. Now, this bacterium is not going to really lose out to anything because it doesn't have to compete with anything else. For example, if there is a person who is not particularly competent at something, uh, and that person will probably not do well in an environment which is highly competitive, where there are others with whom this person has to compete. So this person will basically not be successful in that environment. But this same person, if this person is going to go on an island where this person may be the only person who even has that very limited skill, and there is absolutely nobody for this person to compete with, the person will be totally fine. Now, when that happens, when this sort of suboptimal organism is isolated in a certain environment with nothing for it to compete with, it is under what is called a population bottleneck, right? Where there is basically the space narrows and there isn't too many things which are here. Under these conditions, this bacterium can lose any gene as long as losing it doesn't kill it, right? It may grow slower, it's okay, and as long as it doesn't kill me, I can lose it. So under these pressures, bacterial genomes would tend to become smaller and smaller. You can imagine such population bottlenecks to arise when a free-living bacterium becomes an endosymbiont within a certain larger eukaryotic cell, for example, where it is not really competing with any other bacterium. It can lose things and it can also take things, for example, over time. It can also become more and more dependent on the host, because the host can also provide materials that it can lose. And a classical example of yeah, there is that. A classical example of such process in action is the genome of Mycobacterium leprae. Right, this is the bacterium which causes leprosy. Now, this is a fairly classical genome plot where here is the circular genome. Most bacterial genomes are circular, and then they draw various tracks, right? So the outermost two tracks, let's say, are indicate points at which genes are found on one strand or the other. So you see that there are a lot of genes in this genome. Now look at these two tracks where there are gray lines. Now, these gray lines indicate genes which have become pseudogenes. Pseudogenes means these are degraded genes that no longer function as genes. Wow. If you take any bacterial genome, you may maybe not see more than 10, 15 pseudogenes. But here you have about 4,000 genes, of which looks like probably half of all these genes are pseudogenes. Now, that is clearly an indication a process of genome decay or genome degradation. That is, it started off with a genome with a variety of capabilities. It's now starting to lose these capabilities, and that is because Mycobacterium leprae is a very slow growing bacterium, and it can basically infect only as you know, the nervous system. It can't really survive anywhere else, and it's a super slow growing bacterium, much slow growing than its sister Mycobacterium tuberculosis, and which doesn't have so many pseudogenes. So here is an example of genome degradation in action. If you're interested in other genomes where you see similar things, you can look at the genome of Rickettsia. Rickettsia prowazekii, which is considered by many as the closest extant bacterial ancestor or relative of the mitochondria. So, degradation processes that inform us something about the evolution of Rickettsia. This genome can tell us something about how the mitochondria became what they are today. And genome reduction can also happen through a process of streamlining, which is the complete opposite of this kind of population bottlenecks. Now, a classical example, I'm not going to talk too much about this, but I would encourage you to go and take a look at the genome of this bacterium called Pelagibacter ub. So this is a highly streamlined genome, and it in fact is accounts for a very large fraction of the total microbial biomass on the planet. This is probably the single most dominant bacteria on the planet. It's an ocean bacterium. Ocean, it's also kind of nutrient poor, and what this has done is to reduce its genome, not by loss of selection coming under a population bottleneck, but by increased selection for optimizing its growth in a nutrient poor environment. And these are things that I would suggest that you look at for as your self study. So, are there any questions at this stage?
No, sir. Please go ahead, sir.
Okay. So, as I mentioned at the beginning, the fact that I have horizontal gene transfer indicates that a tree-like evolutionary scenario is not totally appropriate to a bacterial evolution, beyond saying, okay, here is a species and so on. So here I have vertical inheritance being indicated. For example, in this case, okay, I'll use a different color. So in this case, from this over here, so these are obviously belong to the same vertical descent family, and the same time I also see these red lines which indicate horizontal transfer from very different very different families all the way into this family. So here I really do not see an evolutionary picture that is can be cleanly represented by a normal tree. Okay. So how am I doing for time?
Yes, sir.
So
Continue, sir.
do I have time?
Yes, sir. You have time, sir.
Okay. Okay. So now what we talked about were all fairly um large evolutionary changes, horizontal gene transfer. And gene deletion processes sometimes can just have a whole cassette that is being lost. But there is always, for example, we do see this in, say, pseudogenization, where you have small mutations accumulating. Now, there are many small mutations that happen, and though we did mention in, earlier, that a lot of bacterial evolution happens through horizontal gene transfer. In organisms such as Mycobacterium tuberculosis, a lot of antibiotic resistance. Now they've heard about MDR-TB, XDR-TB, some alpha-beta TB, and in these organisms, all these antibiotic resistance evolution typically happens by point mutations happening in certain bases and resulting in very rapid environmental change. So rapid environmental change can happen not only by horizontal transfer, where I have a whole new pathway coming into the gene, but also by point mutations. And this fairly worrisome picture is caused by a combination of both of these phenomena. So here you see a timeline, right, in years, and on top there are on both sides, top and bottom, various antibiotics and classes of antibiotics are listed, and then there is a line which maps to a certain a period in time. The lines on top of this timeline, so this area, mention the dates at which these antibiotics were introduced into clinical practice. Okay, sulfonamides introduced in 1930, 1940. Penicillin obviously introduced sometime during the Second World War. Same with streptomycin, and here we see some of our newer antibiotics like daptomycin. At the bottom, you see the dates in which clinical resistance to this antibiotic was first observed. Sulfonamides introduced here, and then resistance discovered here. And we must also note that if you see a long delay in the development of a resistance, it could be a combination of both a delay in our ability to recognize that resistance is developed, as well as some biochemical aspect of this organism and the antibiotic, which can potentially delay the development of resistance. And you can see daptomycin introduced in the early 2000s, maybe 2004, 2003, something like that, and already within a year, clinical resistance observed. Linezolid 2000, and again 2003, 2004 resistance was discovered. Now, this also tells us that bacteria can evolve very rapidly. For example, in the lab, you can take E. coli and treat it with low concentrations of rifampicin, the antibiotic, which is used even as a first-line therapy against tuberculosis. And within 24 hours, you can isolate E. coli, which are very strongly resistant to this antibiotic. And this is something that is happening very rapidly just through point mutations. For example, when I talk about rifampicin resistance in the laboratory, I don't allow any scope for horizontal gene transfer, and within 24 hours, I have a resistance developing just because there are one or two mutations that have come up and were selected under this rifampicin stress. And when we're talking about evolution of a bacterium towards a certain trait in the laboratory, this is something that you should look up. This is very famous experiment that started in the 1980s. In the 1980s by Richard Lenski, and this experiment is still going on. What Richard Lenski did, he's at the University of Michigan. What he did was to take a few lines of E. coli, and then he put them as usual in a flask, and then he would let it grow till the end of exponential phase, and then take a small volume out and put it back into fresh medium. Keep doing this every day, and at certain points he'll be freezing the cells as a record, and he's been doing that since the 1980s till today. And over this time in the laboratory, E. coli has undergone um, evolution for I think now it's about I think 60,000 generations or so. 60,000 human generations would be would take forever, and we even reached 60,000 generations already in these large numbers of years. Probably no. 60,000 generations of humans, I don't think we have. We don't have. But we have 60,000 generations of these bacteria growing in these flasks. And over time, what Richard Lenski has been able to do is to discover new abilities being acquired by these bacteria. And these were abilities that were thought not to be within the range of abilities that E. coli would have. And one of the most classical examples that you will see would be the ability of E. coli to use citrate, which you would know is a key metabolite in the tricarboxylic acid cycle, as a carbon source. And then initially, of course, in the early years, till the late 2000s, Richard Lenski's experiments were largely low throughput. He would observe a phenotype and then try and make some hypotheses, try and find out what genetic mutation might have happened. But over the last 10 years or so, he's been able to use all these deep sequencing and next generation sequencing technologies to to know the genomes of these various evolved mutations that he's been able to observe. And this is a treasure trove for us. I'm a bioinformatician. Though the lab has done some experimental work, I'm primarily a bioinformatician. And these kinds of genome data, exactly what we would need to try and ask new questions with the new data. And to do so, all that one needs is a computer, right? And a good question to ask, and some skills in programming, know some probability and statistics and so on, and very little money. You just have to pay for a computer which will last many years, and pay for the electricity to run the computer, and then good to go. So there is a lot of interesting science that can emerge from all these data, not just from Richard Lenski's experiment, but also with all the hundreds of thousands of bacterial genomes that we have with us today. All that we need to have is the willingness to go and ask questions, have some questions to ask, and everything that is there about bacteria, a large number of things that is unknown about bacteria are things that we can answer right now at home with a computer and internet and electricity. And all of these data are also freely available, public resource. You can download them. You don't have to pay anything to anybody. You don't have to ask, even ask the person who created these data, can I use this data? You don't have to do that. It is all publicly available. It's part of the open data movement. And just go ahead and do it. There should be hardly anything that should stop us from doing this. I'd like to stop here, unless there are specific questions that you have that I might that might help me take this into a different direction, if that is what you would prefer.
Sir, as of now, there are no questions. I have requested participants to drop in Q&A section and chat box also.
Sure.
You have 10 more minutes, sir. You can proceed, sir.
Okay, so now what we've talked about so far is bacterial evolution by changing genes. And this is a process of adaptation, and these things take time because clearly multi-generational. Now, if I have to adapt within a single generation. Here is a single cell that needs to adapt to a changing environment before it can divide and transmit its genetic content into the next, with or without modifications. The next generation is by altering gene expression states, right? What and how does bacterium do that is by using a variety of transcription factors. For example, your molecular biology, you would have studied the lac repressor. This is a protein that binds to the DNA upstream of a gene, and when it is bound to the DNA, it blocks the expression of this gene. Now, when the nutrient, that is in this case lactose, is available, this molecule will come and bind to this protein. And when this molecule is bound to this protein, lactose, this protein leaves the DNA, and that allows this set of genes to be expressed. And these genes would obviously code for proteins that can utilize lactose as a carbon source and provide energy to the cell. So here you have gene expression control that is acting on demand. When there is a requirement, switch on the related required gene. And why do you need to do this? Why do you not just have everything expressed at all times? The answer is simple. Expressing genes is expensive. It will make use of resources that the bacterium would have. And secondly, certain gene functions may interfere with the activities of certain other gene functions. So when a certain gene function is necessary, then it is important to keep the antagonistic gene function shut down, right? So this is the LAC system, and typically when one looks at gene regulatory networks, one would represent this as in a simple network-like representation. Here is a gene, and here is the protein. Here is the nutrient that activates this or that takes off this protein. Now this is a protein which can bind to this DNA, so it should actually be a repressor. So this would be repressing this gene, and this is a repressor of this gene, and this is a simple representation. And one could have a variety of information depending on what is available on these arrows. It could be a 1-1 type thing, activation versus repression, or you could have biochemical information such as the dissociation constants and so on, depending on the kinds of analysis that one wants to do. Now, let's just take this LAC system, and you'll see that it is slightly more complicated than that. You just do not have a single repressor, and you also have a second regulator, which also comes and plays a role. Now, this second regulator is something which is called a global regulator. And this is something which actually has more than 300 different targets. So what is this? So E. coli obviously has a preferred carbon source, for example, that is glucose, and it will utilize lactose or any other alternate carbon source only when glucose is not present. Now, what this does, this major regulator does, is to make sure that any alternate carbon source is utilized only when the major carbon source of glucose is not present. That means this is something, this protein will have to respond in some way to the levels of the major carbon source, and simultaneously inform this news that there is glucose available or not available to a whole variety of downstream genes. So now you are already seeing that this network is starting to and if I take all the data that are available for the regulatory network for E. coli, I'll see some really related by these genes, and then you have on-demand regulators like these, which have a few targets, each of these lines indicating a regulatory interaction, and then here are these very, very large red blobs in the middle, which have a lot of targets, as you can see from the large number of lines that are radiating out of these lines, and these are global regulators. Now, this overall transcription control has a combination of on-demand specific regulators and this global homeostasis under a wide range of parameters being maintained by what are known as these global regulators of gene expression. Now, taken together, adaptation can occur at multiple time scales. Within a generation, you have adjustments coming out of gene expression control, and over time, even from a few generations, thousands of generations, and E. coli has been around now for 100 million years. So, a lot of evolution has happened, and these are mechanisms which will involve genetic changes. And going forward, some of our own labs work has been interested in seeing how genetic evolution would affect gene expression changes and so on. But that would be for another day. So I would like to stop here and take questions, and also thank you for listening.
Thank you very much, sir, for a wonderful session. We have two questions from the participants.
Yes.
Does anyone have some same microbes in body?
Sorry?
Same microbes in body.
Oh, yeah. So there is now a lot of literature on the human microbiomes and so on. And what we do see is that there are certain kinds of bacteria which define, say, the human microbiome, and this may be present in a certain kinds of organisms, but not be commonly found in other kinds of organisms. And there are also rarer, and these are very, very predominant. There are large numbers of these bacteria present, and there are also rarer components of the human microbiome which can also be more variable. So, in fact, there is this bacterium whose name was a source of great entertainment for us when we were in college. It was called Bacteroidetes theta iota omicron. So that is what it was called, Bacteroidetes theta iota micron. And it turns out that these Bacteroidetes, this genus of bacteria, are the top one or top two in terms of their numbers in the human gut. So now there is a lot of literature just studying this genus of bacterium. So we do share a large component of the microbiome, but there could also be bacteria that are fairly unique. But is it unique enough to be like a fingerprint and so on? I don't know.
Thank you, sir. There is one more question. What happens when bacteria are exposed to poor nutrients and poor condition?
To exposed to
Poor nutrients and poor condition.
Sure. So now they obviously have to adapt, right? So there is an interesting balance between wanting to grow and wanting to respond to stress. Now, it turns out that in many bacteria there is a trade-off. If you're responding to stress, you're not growing, and if you're growing, then you're not very good at responding to stress. So many molecular mechanisms that bacteria have will help the bacterium in response to conditions, for example, nutrient starvation or various other kinds of suboptimal conditions, to adjust this balance between growth and stress response, and that would be a predominant mechanism by which this would develop. And say if you have a fast-growing bacterium that is exposed to a low nutrient condition for a prolonged period of time, then you would also expect to see genetic mutations which will come in and which would eventually target these kinds of balances. So one example that I would ask you to try and look up in the literature is what is called
Yes, sir.
GASP in E. coli.
Yes. Yes.
This is called growth advantage in stationary phase. Okay, so some of the literature on this will probably tell you something more about these kinds of adaptations. And I would also point you to the works of an author called Ferenczi, who has talked quite a lot about this balance.
Thank you very much, sir. Now, before concluding, I request our CEO, Dr. M. Ramesh, sir, to speak few words.
Students got a lot of information to broaden the horizon of their knowledge. Thank you. And we have a good training center at KSTA. We will most probably organize workshops